Welcome back to part 2 of our Ethiopian University Entrance Examination Biology Revision series. In this section, we will discuss questions related to photosynthesis and cellular respiration. For each question, we will first read the question, identify the correct answer, and then explain the scientific concept behind it in a simple and exam-focused way. Let's begin. Question number 1. In a hypothetical scenario where photosynthesis ceased to occur on the Earth, the long-term consequences on the continuity of life would be: A. Remained unaffected since other sources of energy exist. B. Collapse that could lead to mass extinction. C. Balanced between carbon dioxide and oxygen appropriately. D. Disordered although animal life would flourish. The correct answer is B. Photosynthesis is the foundation of nearly all life on Earth. Through photosynthesis, plants produce glucose, which serves as food for ecosystems, and oxygen, which supports aerobic life. If photosynthesis stopped completely, no new food would be produced. Food chains and food webs would eventually collapse. Oxygen levels would continuously decrease. Carbon dioxide levels would rise. As a result, most living organisms, including humans and animals, would ultimately die. Therefore, the long-term consequence would be a collapse of life leading to mass extinction. Question number 2. Which of the following is the difference between substrate-level phosphorylation and oxidative phosphorylation? Substrate-level phosphorylation: A. Occurs in the inner mitochondrial membrane while oxidative phosphorylation occurs in the cytosol. B. Occurs during glycolysis and Krebs cycle while oxidative phosphorylation occurs during the electron transport chain. C. Has the same source of energy as oxidative phosphorylation. D. Occurs in aerobic respiration while oxidative phosphorylation occurs in anaerobic respiration. The correct answer is B. There are two major ways cells produce ATP. The first is substrate-level phosphorylation. In this process, ATP is formed directly when a phosphate group is transferred from a substrate molecule to ADP. This occurs during glycolysis and the Krebs cycle. The second is oxidative phosphorylation. This process takes place in the electron transport chain located on the inner mitochondrial membrane. It uses energy released from electron transfer to generate a proton gradient, which drives ATP synthase to produce ATP. Most ATP in aerobic respiration is produced through oxidative phosphorylation. Question number 3. Which one of the following is correct about the energy liberation phase of glycolysis pathway? It: A. Hydrolyzes two ATP molecules and raises the free energy of glucose. B. Breaks 6 carbon molecules into 2 glyceraldehyde, 3-phosphate molecules. C. Releases 1 water molecule and dihydroxyacetone phosphate molecule. D. Produces 4 ATP, 2 NADH and 2 molecules of pyruvate. The correct answer is D. Glycolysis occurs in 3 major phases. The first phase is the energy investment phase, where 2 ATP molecules are used. The second phase is the splitting phase, where glucose is divided into 2 3-carbon molecules. The third phase is the energy liberation or payoff phase. During this stage, ATP and NADH are produced. The products include 4 ATP molecules, 2 NADH molecules, 2 pyruvate molecules, and 2 water molecules. Therefore, option D correctly describes the energy liberation phase. Question number 4. In a complete oxidation of a glucose molecule, how many net ATP molecules are produced by substrate-level phosphorylation? A. 2. B. 4. C. 8. D. 10. The correct answer is B. Substrate-level phosphorylation occurs only during glycolysis and the Krebs cycle. Glycolysis produces a net gain of 2 ATP molecules. The Krebs cycle produces 2 ATP molecules per glucose. Adding these together gives a total of 4 ATP molecules. All remaining ATP is generated through oxidative phosphorylation in the electron transport chain. Therefore, the correct answer is 4 ATP. Question number 5. What is the difference between aerobic and anaerobic respiration? A. Aerobic respiration ends with different products while the same product results in anaerobic respiration. B. Proteins are the main respiratory substrate for both aerobic and anaerobic respiration. C. Aerobic respiration produces much more energy than anaerobic respiration. D. Glycolysis is the first pathway for aerobic while Krebs cycle is for anaerobic respiration. The correct answer is C. Aerobic respiration requires oxygen. It completely oxidizes glucose into carbon dioxide and water. As a result, it produces approximately 36 ATP molecules per glucose. Anaerobic respiration occurs without oxygen. It only partially breaks down glucose and produces about 2 ATP molecules per glucose. Because aerobic respiration completely oxidizes glucose, it releases much more energy. That is why option C is correct. Question number 6. Which one of the following descriptions about glycolysis is correct? It is the process A. Of breaking down of a glucose molecule to 2-pyruvates. B. That takes place in the cytosol with high demand of oxygen. C. That produces a net of 4 ATP through substrate-level phosphorylation. D. Of producing acetyl-CoA from pyruvate by releasing carbon dioxide. The correct answer is A. Glycolysis is the first stage of cellular respiration. It occurs in the cytoplasm, also called the cytosol. One 6-carbon glucose molecule is broken down into 2 3-carbon pyruvate molecules. This process does not require oxygen. In addition, glycolysis produces 2 ATP molecules net and 2 NADH molecules. Therefore, option A is correct. Question number 7. Which one of the following statements properly describes the structure of ATP? It is made up of A. 2 adenine bases attached to a deoxyribose sugar and 2 phosphate groups. B. 1 adenine base attached to a ribose sugar and 2 phosphate groups. C. 2 adenine bases attached to a hexose sugar and 3 phosphate groups. D. 1 adenine base attached to a pentose sugar and 3 phosphate groups. The correct answer is D. ATP stands for adenosine triphosphate. ATP consists of 3 components. 1 adenine base. 1 ribose sugar, which is a 5-carbon pentose sugar. And 3 phosphate groups. The bonds between the phosphate groups store usable energy for the cell. Therefore, option D correctly describes ATP structure. Question number 8. During oxidative phosphorylation, ATP is produced by combining ADP and inorganic phosphate using energy released from A. Decarboxylation of citrate to oxaloacetate. B. Transfer of electrons from NADH and FADH2 to oxygen. C. Dehydrogenation of pyruvate to acetyl-CoA. D. Transport of electrons from complex 4 to complex 1. The correct answer is B. During oxidative phosphorylation, NADH and FADH2 donate electrons to the electron transport chain. As electrons move through the chain, energy is released. This energy pumps hydrogen ions across the inner mitochondrial membrane. The resulting proton gradient drives ATP synthase, which produces ATP. Oxygen acts as the final electron acceptor and combines with electrons and hydrogen ions to form water. Therefore, ATP production is ultimately powered by electron transfer from NADH and FADH2 to oxygen. Question number 9. Which one of the following comparisons between substrate level and oxidative phosphorylation is correct? During substrate level phosphorylation, A. Protons are pumped into the intermembrane space, but in oxidative phosphorylation they are pumped into the cytosol. B. Oxygen is the final electron acceptor, but in oxidative phosphorylation NAD is the electron acceptor. C. ATP is made in the cytosol and matrix, but in oxidative phosphorylation it is made on the inner membrane of mitochondria. D. 34 ATP molecules are produced, but in oxidative phosphorylation 4 ATP molecules are produced per glucose molecule. The correct answer is C. Substrate-level phosphorylation occurs during glycolysis in the cytoplasm and during the Krebs cycle in the mitochondrial matrix. ATP is produced directly by transferring a phosphate group to ADP. Oxidative phosphorylation occurs on the inner mitochondrial membrane. It depends on the electron transport chain, chemiosmosis, and ATP synthase. Therefore, option C correctly compares the locations of ATP production. Question number 10. Which one of the following determines the cause and effect of lactate production during physical exercise? The demand for A. Lower pH in muscle tissues leads to fermentation of respiratory substrates leading to muscle fatigue. B. More energy causes muscle cells to ferment respiratory substrates leading to muscle fatigue. C. An easily absorbable molecule leads to the production of lactate that results in muscle fatigue. D. A substrate that can easily release energy leads to the production of lactate which results in muscle fatigue. The correct answer is B. During vigorous exercise, muscles require large amounts of ATP. Sometimes oxygen delivery cannot keep up with this high demand. When oxygen becomes limited, muscle cells switch from aerobic respiration to anaerobic respiration. Pyruvate is converted into lactate, allowing glycolysis to continue producing ATP. As lactate accumulates, muscle fatigue and discomfort increase. Generally, high energy demand and reduced oxygen availability lead to lactate fermentation, which lead to muscle fatigue. Therefore, option B is the correct answer. Question number 11. In aerobic cellular respiration, A. Pyruvate enters into the mitochondria for further stages. B. Glycolysis takes place in the mitochondria. C. Krebs cycle takes place in the cytoplasm. D. Electron transport chain takes place in the cytoplasm. The correct answer is A. Cellular respiration occurs in several stages. Glycolysis takes place in the cytoplasm and produces pyruvate. The pyruvate molecules then enter the mitochondria, where the link reaction, Krebs cycle, and electron transport chain occur. Therefore, pyruvate entering the mitochondria is the correct statement. Question number 12. Which one of the following processes of aerobic respiration takes place in the Krebs cycle? A. 2-carbon group from acetyl coenzyme A reacts with a 4-carbon compound to form citrate. B. Fructose 1,6-bisphosphate is split into 2 molecules of the 3-carbon sugar GP. C. Hydrogen atoms carried by reduced NAD and FAD are released and split into protons and electrons. D. 2 molecules of ATP are used to phosphorylate each molecule of glucose. The correct answer is A. The Krebs cycle begins when acetyl-CoA, which contains 2 carbons, combines with oxaloacetate, a 4-carbon molecule. This reaction forms citrate, a 6-carbon compound. Question number 13. Which of the following processes takes place when lactate is produced? A. Protons accumulate in the intermembrane space. B. Reduced NAD supplies hydrogen and becomes oxidized itself. C. NAD and FAD are regenerated from reduced NAD and FAD. D. ATP is produced through oxidative phosphorylation. The correct answer is B. During lactate fermentation, pyruvate receives hydrogen from NADH. As a result, pyruvate is converted into lactate. At the same time, NADH loses hydrogen and becomes oxidized back to NAD. This regenerated NAD allows glycolysis to continue producing ATP. Therefore, option B is correct. Question number 14. What happens during the electron transport system of respiration? A. Citrate loses a carbon atom to form a 5-carbon compound and carbon dioxide. B. A 5-carbon compound is further decarboxylated to form a 4-carbon compound. C. A 4-carbon compound undergoes molecular transformation to generate oxaloacetate. D. Hydrogen atoms carried by FADH2 and NADH are released and split into protons and electrons. The correct answer is D. In the electron transport chain, NADH and FADH2 donate hydrogen. These hydrogen atoms separate into protons and electrons. The electrons move through a series of carrier proteins, releasing energy. That energy is used to pump protons and establish a proton gradient for ATP production. Therefore, option D is the correct answer. Question number 15. The hydrogen ions that are used to turn the rotor of ATP synthase come from A. Phosphorylation of ADP. B. Hydrolysis of ATP. C. Oxidation of NADH and FADH2. D. Reduction of NADH and FADH2. The correct answer is C. When NADH and FADH2 are oxidized in the electron transport chain, they release electrons and hydrogen ions. The hydrogen ions accumulate in the intermembrane space. As they flow back through ATP synthase, the enzyme rotates and synthesizes ATP. Therefore, the hydrogen ions originate from the oxidation of NADH and FADH2. Question number 16. What triggers fermentation during exercise? A. Shortage of oxygen. B. Overactivity of respiratory enzymes. C. Production of ATP beyond cell demand. D. Presence of excess glucose. The correct answer is A. During intense physical activity, muscles consume oxygen rapidly. When oxygen delivery cannot meet demand, aerobic respiration slows. To continue producing ATP, cells switch to anaerobic respiration. Fermentation regenerates NAD and allows glycolysis to continue. Therefore, oxygen shortage is the trigger for fermentation. Question number 17. Which of the following processes explains lactate production during anaerobic respiration in animal cells? A. Oxidation of pyruvate. B. Decarboxylation of pyruvate. C. Phosphorylation of pyruvate. D. Reduction of pyruvate. The correct answer is D. In animal cells, pyruvate accepts hydrogen and electrons from NADH. Gaining hydrogen or electrons is called reduction. As a result, pyruvate is converted into lactate. Therefore, lactate production occurs through reduction of pyruvate. Question number 18. Which one of the following descriptions holds true for the link reaction of respiration? A. Pyruvate reacts with coenzyme A to form acetyl coenzyme A. B. Glucose is converted to pyruvate through a chain of reactions. C. Hydrogen ions and ATP are synthesized. D. Reduced NAD and reduced FAD are produced. The correct answer is A. The link reaction connects glycolysis with the Krebs cycle. During this process, pyruvate enters the mitochondrion. One carbon atom is removed as carbon dioxide. NAD is reduced to NADH. The remaining two carbon fragment combines with coenzyme A to form acetyl-CoA. Therefore, option A correctly describes the link reaction. Question number 19. Which event of cellular respiration takes place in the cytoplasm of eukaryotic cells? A. Production of pyruvate from glucose. B. Reaction of acetyl-CoA with oxaloacetate. C. Production of ATP through chemiosmosis. D. Reaction of pyruvate with coenzyme A. The correct answer is A. Only glycolysis occurs in the cytoplasm. During glycolysis, glucose is broken down into pyruvate. The link reaction and Krebs cycle occur in the mitochondrial matrix, while chemiosmosis occurs on the inner mitochondrial membrane. Therefore, option A is correct. Question number 20. Suppose 2 moles of glucose are burned aerobically. How many moles of ATP can be produced by substrate-level phosphorylation in the Krebs cycle? A. 4. B. 6. C. 10. D. 8. The correct answer is A. One glucose molecule produces 2 ATP through substrate-level phosphorylation during the Krebs cycle. Therefore, 2 glucose molecules will produce 4 ATP molecules. Remember that the question asks only about ATP produced in the Krebs cycle, not total ATP production. Question number 21. During alcoholic fermentation, carbon dioxide is released when A. Acetaldehyde is reduced. B. Pyruvate is oxidized. C. Pyruvate is decarboxylated. D. Ethanol is oxidized. The correct answer is C. Alcoholic fermentation commonly occurs in yeast cells. The process takes place in 2 major steps. First, pyruvate loses a carbon atom in the form of carbon dioxide, producing acetaldehyde. This step is called decarboxylation. Second, acetaldehyde is reduced by NADH to form ethanol. Since carbon dioxide is released during the first step, the correct answer is option C. Question number 22. During anaerobic respiration, reduced NAD is required to A. Split pyruvate to carbon dioxide and acetaldehyde. B. Reduce pyruvate to lactate. C. Convert lactate into pyruvate. D. Reduce pyruvate to acetyl-CoA. The correct answer is B. During anaerobic respiration in animal cells, pyruvate accepts hydrogen from NADH. This converts pyruvate into lactate. At the same time, NADH becomes oxidized back to NAD. The regenerated NAD allows glycolysis to continue producing ATP. Therefore, reduced NAD is required to reduce pyruvate to lactate. Question number 23. Why do we get tired and fatigued during heavy exercise such as long-distance running? It is due to A. Accumulation of carbon dioxide. B. Accumulation of lactic acid. C. High rate of aerobic respiration. D. Shortage of glucose. The correct answer is B. During prolonged or intense exercise, oxygen supply may not meet the demand of working muscles. As a result, muscles begin anaerobic respiration. Pyruvate is converted into lactate. As lactate accumulates, muscle discomfort and fatigue increase. Therefore, the accumulation of lactic acid is the main reason for muscle fatigue during strenuous exercise. Question number 24. Which of the following products are formed in anaerobic respiration of one glucose molecule in animal cells? A. 2-lactate, 2-carbon dioxide, and 2-ATP. B. 2-ethanol, 2-carbon dioxide, and 2-ATP. C. 2-ethanol, 2-NAD, and 2-ATP. D. 2-lactate, 2-NAD, and 2-ATP. The correct answer is D. In animal cells, anaerobic respiration occurs through lactate fermentation. Glycolysis first produces pyruvate, ATP, and NADH. The pyruvate is then reduced to lactate. During this process, NADH is oxidized back to NAD. The final products are 2 lactate molecules, regenerated NAD, and a net gain of 2 ATP molecules. Question number 25. Which stage of cellular respiration is correctly paired with its site? A. Glycolysis, Christie. B. Pyruvate oxidation, cytoplasm. C. Chemiosmosis, outer membrane. D. Krebs cycle, matrix. The correct answer is D. Different stages of respiration occur in different locations. Glycolysis occurs in the cytoplasm. Pyruvate oxidation occurs in the mitochondrial matrix. The Krebs cycle also occurs in the mitochondrial matrix. Chemiosmosis occurs on the inner mitochondrial membrane. Therefore, the correctly matched pair is Krebs cycle and matrix. Question number 26. What happens during the process of the electron transport chain? Protons. A. Diffuse against the concentration gradient through ATP synthase. B. Build up more in the matrix than the intermembrane space. C. Diffuse down the concentration gradient to reduce NAD. D. Build up more in the intermembrane space than the matrix. The correct answer is D. As electrons move through the electron transport chain, energy is released. This energy pumps hydrogen ions from the mitochondrial matrix into the intermembrane space. As a result, a proton gradient is formed. The concentration of protons becomes much higher in the intermembrane space than in the matrix. This gradient later drives ATP synthesis. Therefore, option D is correct. Question number 27. Question number 27. Identify the correct statement about the role of NAD and FAD during aerobic respiration. They A. Are the terminal electron acceptors of the electron transport chain. B. Carry hydrogen atoms that will be split into protons and electrons. C. Oxidize intermediate products of cellular respiration. D. Are electron carriers that pump protons into the intermembrane space. The correct answer is B. NAD and FAD function as hydrogen carriers. During respiration they become NADH and FADH2. These molecules transport hydrogen to the electron transport chain. There, hydrogen is separated into protons and electrons. The electrons travel through the chain while the protons contribute to the proton gradient. Therefore, option B correctly describes their role. Question number 28. Which one of the following options represents the correct catabolic pathway of the given respiratory substrate? A. Lipids fatty acids acetyl-CoA TCA cycle. B. Polysaccharide glucose acetyl-CoA pyruvate TCA cycle. C. Nucleic acids amino acids nucleotide acetyl-CoA TCA cycle. D. Proteins ammonia amino acid pyruvate TCA cycle. The correct answer is A. Lipids are first broken down into fatty acids and glycerol. Fatty acids undergo beta-oxidation. This process produces acetyl-CoA. Acetyl-CoA then enters the Krebs cycle, also known as the TCA cycle. Question number 29. In which part of the mitochondrion does the Krebs cycle take place? A. In the matrix. B. On the inner membrane surface. C. On the outer membrane surface. D. In the intermembrane space. The correct answer is A. The mitochondrial matrix contains the enzymes required for the Krebs cycle. This is where acetyl-CoA is oxidized and energy-rich molecules such as NADH and FADH2 are produced. Question number 30. How many ATP molecules are produced during the complete cellular respiration of one glucose molecule? A. 2. B. 4. C. 18. D. 36. The correct answer is D. Glycolysis produces 2 net ATP. The Krebs cycle produces 2 ATP. Oxidative phosphorylation produces approximately 32 ATP. This gives a total of about 36 ATP molecules from one glucose molecule. Therefore, the correct answer is 36 ATP. Question number 31. The different steps for complete aerobic respiration are: A. Glycolysis and oxidative phosphorylation. B. Glycolysis and Krebs cycle. C. Glycolysis, Krebs cycle, and terminal oxidation. D. Krebs cycle and terminal oxidation. The correct answer is C. Complete aerobic respiration consists of several interconnected stages. First, glycolysis breaks down glucose into pyruvate. Next, pyruvate is processed and enters the Krebs cycle. Finally, terminal oxidation, which includes the electron transport chain and chemiosmosis, generates most of the ATP. Although the link reaction is not listed in the options, option C is the most complete answer available. Therefore, option C is correct. Question number 32. Which of the 4 stages in the aerobic respiration of glucose does not produce ATP? A. Glycolysis. B. The link reaction. C. Krebs cycle. D. Electron transport and chemiosmosis. The correct answer is B. The link reaction serves as a bridge between glycolysis and the Krebs cycle. During this stage, pyruvate is converted into acetyl-CoA. Carbon dioxide is released. NAD is reduced to NADH. However, no ATP is produced. In contrast, glycolysis, the Krebs cycle, and oxidative phosphorylation all generate ATP. Question number 33. Which one is first inhibited if a cell contains excess ATP? A. Krebs cycle. B. Oxidative phosphorylation. C. Glycolysis. D. Electron transport. The correct answer is C. Cells carefully regulate ATP production. When ATP levels become high, the cell reduces unnecessary energy production. One important enzyme in glycolysis, phosphofructokinase, is inhibited by high ATP concentrations. As a result, glycolysis slows down first. This prevents wasteful breakdown of glucose when energy is already abundant. Question number 34. Fermentation of glucose by yeast produces: A. Lactic acid and oxygen. B. Ethyl alcohol and oxygen. C. Lactic acid and carbon dioxide. D. Ethyl alcohol and carbon dioxide. The correct answer is D. Yeast cells carry out alcoholic fermentation. In this process, pyruvate is converted into acetaldehyde, releasing carbon dioxide. Acetaldehyde is then reduced to ethanol. As a result, the final products are ethanol, also called ethyl alcohol, and carbon dioxide. Therefore, option D is correct. Question number 35. In cellular respiration, the carriers for the electron transport system are located: A. Within the stroma of the chloroplast. B. On the cristae of mitochondria. C. Within the matrix of mitochondria. D. Within the cytoplasm of a cell. The correct answer is B. The electron transport chain is located on the inner mitochondrial membrane. The folds of this membrane are called cristae. These folds increase surface area and provide space for electron carriers, ATP synthase, and other proteins involved in ATP production. Therefore, the electron transport carriers are located on the cristae. Question number 36. Which of the following pathways will use coenzyme A during aerobic cellular respiration? A. Glycolysis. B. Link reaction. C. Krebs cycle. D. Calvin cycle. The correct answer is B. During the link reaction, pyruvate enters the mitochondrion. One carbon atom is removed as carbon dioxide. NAD is reduced to NADH. The remaining two carbon fragment combines with coenzyme A to form acetyl-CoA. Because coenzyme A directly participates in the link reaction, option B is the correct answer. Question number 37. How many times does the Krebs cycle turn per glucose molecule broken down? A. 5 times. B. 4 times. C. 2 times. D. 3 times. The correct answer is C. One glucose molecule produces 2 pyruvate molecules through glycolysis. Each pyruvate is converted into one acetyl-CoA molecule. Each acetyl-CoA enters the Krebs cycle once. Therefore, one glucose molecule results in 2 turns of the Krebs cycle. Question number 38. Which one of the following metabolic pathways is common in aerobic and anaerobic respiration? A. Glycolysis. B. Chemiosmosis. C. The citric acid cycle. D. Oxidative phosphorylation. The correct answer is A. Both aerobic and anaerobic respiration begin with glycolysis. Glycolysis occurs in the cytoplasm and does not require oxygen. The Krebs cycle, oxidative phosphorylation, and chemiosmosis occur only during aerobic respiration. Question number 39. Which of the following processes produces the most ATP per molecule of glucose oxidized? A. Aerobic respiration. B. Anaerobic respiration. C. Alcoholic fermentation. D. Lactic acid fermentation. The correct answer is A. Aerobic respiration completely oxidizes glucose into carbon dioxide and water. Because it includes glycolysis, the Krebs cycle, and oxidative phosphorylation, it generates approximately 36 ATP molecules per glucose. Anaerobic respiration and fermentation produce only about 2 ATP molecules. Therefore, aerobic respiration produces the greatest amount of ATP. Question number 40. The following diagram represents some event that takes place in plant cells. In which organelle would the above events most likely occur? A. Lysosome. B. Ribosome. C. Mitochondrion. D. Chloroplast. The correct answer is C. The diagram shows sugar entering the organelle, oxygen, O2, entering, carbon dioxide, CO2, leaving, water, H2O, leaving, ADP being converted to ATP. These are the key features of aerobic respiration. These are the key features of aerobic respiration. The organelle responsible for aerobic respiration in plant cells is the mitochondrion.